स्क्रीन बरोबर आहे काही आपण मॅडम मला आहे ना तर त्यांना हे लावायचं बरोबर कॉलर मला एकदा म्हटलं पीडीएफ जरा सध्या शेअर करा, मी कंटिन्यू करतो. पीडीएफ किंवा पीपीटी जी असेल ती मला ती फॉरवर्ड करायला लागते. हर्षल ही जी लिंक आहे ती मला फॉरवर्ड करा आत्ताची, जे लाईव्ह लेक्चर चालू केलं आपण ना पहिली. मला फॉरवर्ड करा YouTube याला WhatsAppला. हां, लाईव्ह आत्ता जे चालू आहे ते. पीपीटी आहे, पीडीएफ आहे. त्या पाठवा सगळ्या. मागील वेळच्या पाठवल्या त्या फॉरवर्ड केल्या स्टुडंट्स. हो. नाही जसं असेल तसं पाठवा, नो प्रॉब्लेम. आपण त्याला एडिट करत नाही.
काय केलेलं ऍड म्हणा तुमचे ब्लँक बोर्ड येतो का इथे? अच्छा. ओके. ओके.
त्यानंतर एक ऍड लाईन पाहिजे. त्यानंतर मागे मागचे मागे पुढे जायचं.
Okay. Fine. Wonderful. For writing purpose. Sure. Thank you so much. This end or this end? White end, na sir? This end, this end. Okay, okay, okay. Sure. So good morning, students. In the last lecture, I guess you are new for this lecture. Last lecture, we have started with the cells, basic structure of cell, basic differentiation between prokaryotic cell, eukaryotic cell. पुढे. And then we have also seen the cell theory. ठीक आहे? सेल थिअरी मला कोण कोण सांगेल लास्ट लेक्चरचे प्रेझेंट स्टुडंट्स? सेल थिअरी कोणी कोणी प्रपोज केली? शेल्डन, शॉन अँड वर्चॉव, राईट? या तीन सायंटिस्ट मिळून काय प्रपोज केलाय? सेल थिअरी. सो सेल थिअरीचं मेन जिस्ट काय आहे? पहिलं काय आहे? एव्हरी लिव्हिंग ऑर्गॅनिझम इज मेड अप ऑफ सेल्स, राईट? Plants are made up of cells, animals are made up of cells, different type of cells. Third is a new cell is formed from the pre-existing cell. ठीक आहे? हे मेन तीन आहेत. आणि चौथं काय आहे? That is cell is the structural and functional unit of the life. We have seen that. So there are three basic fundamental, you can say, postulations of the cell theory. पहिलं काय आहे? Plant and animals, they are made up of cells. Second is what? Every new cell is formed from the pre-existing cell, and cell is the structural and functional unit of life. Okay, we have seen also the basic history of the cell biology, that is how the study of cell, right? How we define cell biology? It is a branch which deals with the study of cell with respect to structure and function. Okay, and we have seen that this particular study of cell involves contribution of lot of different scientists. Okay, we have seen who invented microscope? Antony von Leeuwenhoek, right? Who invented the term cell? Robert Hooke. Okay, how he discovered the term cell? He took a slice of a cork tissue and he observed it under the microscope. Cork tissue is present in which organism? It is present in plants. It's a dead tissue. Okay, and when he observed that particular cells, what he got to know? Yes, we got to know that the organization of that tissue of the cork is in such a way that it has small chambers, right? And he called those small chamber like structure as what? Cellula. And from that term, the term was developed as a cell. Understanding? So this short history we have seen, we have started from Aristotle, how he contributed for this invention of the term cell and the concept of cell, till we have come towards the cell theory. So remember who proposes cell theory? Sheldon, Schwann and Virchow. What is the first postulate of cell theory? Plants and animals, they are made up of multiple different type of cells. Okay. Second postulate, every new cell is formed from the pre-existing cell. And third postulate is what? Cell is a structural and functional unit of life. Now, after that we have moved towards the difference between prokaryotic cell and the eukaryotic cell, right? What do you mean by prokaryotic cell? The cell which lack nucleus or the cell which does not have nucleus is called as what? Prokaryotic cell. What is eukaryotic cell? The cell which is having the true nucleus in its cytoplasm is called as what? Eukaryotic cell, right? So if you see basic differentiation which we have seen between prokaryotic cell so if you see the basic difference between prokaryotic cell and eukaryotic cell, the first differentiation which we have seen is regarding prokaryotic cell. Karyotic cell. Okay, what is the first basic differentiation? I am writing in short. Nucleus is absent. Over here, nucleus is present. First point. In case of prokaryotic cell, okay, where is the DNA present? DNA is naked. I have seen one more concept. The naked DNA which is present in the cytoplasm, that naked DNA is present in which form? It is present in nucleoid form, if you remember. Okay? Over here, what is the second point in case of eukaryotic cell? DNA is present in nucleus. The DNA which is present of the prokaryotic cell, what is the nature of that DNA? The DNA in case of prokaryotic cell is circular. In nucleoid form. Okay, nucleoid structure of that DNA is present. It means what? If this see circular DNA is like this. Okay, so how is the nucleoid structure? If you see a bacterial cell, all right, in that this circular DNA is twisted like this. This is what is the nucleoid structure. So this circular DNA is further, if you can see, it is converted into, or it is, you can say, supercoiled and twisted into a bunch-like structure. This is what is called as nucleoid. And this nucleoid structure is what? It is simply suspended within the cytoplasm. Okay. So this is the main difference between prokaryotic cell and eukaryotic cell. What is the very first difference, which is the main demarcation? In prokaryotic cell, the nucleus is absent. In eukaryotic cell, the nucleus is present. Okay. Now, in case of prokaryotic cell, where the DNA is present, DNA will be present inside the cytoplasm. In which form? In naked form. Okay. Whereas, in case of eukaryotic cell, if you see, the DNA is present where? The DNA is present inside the nucleus, inside the nucleoplasm. Okay. Now, in case of prokaryotic cell, what is the DNA contained? A single circular chromosome. And that single circular chromosome is further supercoiled and twisted into folds, and that structure is called as what? Nucleoid. This is the nucleoid structure. Whereas in case of eukaryotes, if you see, okay, the structure of chromosome in case of eukaryotes is like this. It is a linear chromosome. And if you see the structure of linear chromosome, this is what? This is your centromere. Okay, a small circular structure. And these are what? These are your chromatids. Okay, this is one chromatid, this is another chromatid. So these two chromatids, they are called as what? Sister chromatids. Sister chromatids means what? Chromatids of the same chromosome. They are called as what? Sister chromatids. We also call it in genetics as this is P arm, P arm. This is Q arm. Name of the chromatids. Upper one is P, lower one is Q. And these two are what? Sister chromatids. So in case of eukaryotic cell, if you see this chromosome which is present, that chromosome is in which form? It is in the linear form. It is not circular. Okay. And this particular chromosome structure, it has been associated with a, you can say, combination of histone proteins. All right. Now see, imagine one thing. You have studied the structure of DNA, right? How is the structure of DNA? It is a double-stranded structure, right? One strand, if this strand, it runs in 5' to 3' direction, this will run in 5' to 3' direction, opposite, okay? So this DNA is what? DNA is a free molecule, okay? It is present inside the nucleus. And what it is made up of? It is made up of nucleotides, okay? Adenine, guanine, thymine, cytosine, these are the nitrogen bases which combine to form the nucleotides. All right. So now, what is this chromosome? This is the structure of this highly compacted nature of the DNA. When DNA is not compacted, it looks like this. But when you compact it, okay, to form 1400 nanometer fiber, it looks like this. So what is chromosome in short? It is a compacted form of the DNA. So when I say my body has 46 chromosomes, each and every cell, it means what? I have 46 different DNA molecules. Understanding? When I say a human cell has 23 pairs of chromosome, means what? It has 23 pairs of DNA molecules. Understanding? So one chromosome means what? One DNA molecule, which has been compactedly packed to attain this structure. Okay. So always remember, in case of eukaryotic cell, the chromosome is how it is? Linear. And the complete DNA material, it is present in which form? It is present in chromatin form. We are going to study the structure of chromatin in the upcoming lectures. Okay. So remember, what is chromatin? DNA plus histone protein is called as what? Chromatin. So this DNA plus histone protein combination, it does what? It further leads to the formation of chromatin structure. All right. So in case of eukaryotes, the nucleus contains the DNA in which form? In chromatin form. In case of prokaryotes, the DNA is present in which form? In nucleoid structure form. Understanding? In prokaryotic cell, nucleus is absent. In eukaryotic cell, nucleus is present. In case of prokaryotic cell, the DNA, that is a chromosome, is present in the cytoplasm in the naked form. In which form? Nucleoid structure. In case of eukaryotes, the chromosome is linear. And the DNA contained is present in which form? It is present in the chromatin form inside the nucleus, because it is the eukaryotic cell. Understanding? So now, if you see, we have seen this basic differentiation. First was based upon the nucleus. Second was the arrangement of the DNA. Now, third main difference which comes in this particular eukaryotic and prokaryotic cell differentiation is the ribosomes are very small. Okay. They are of which type? They are of 70S type. In case of eukaryotic cell, the ribosomes, they are large. They are of which type? They are of ATS type. Now you must be confused. What is 70S and what is ATS, right? So now if you see ribosomes, they are mainly manufactured in the eukaryotic cell inside the nucleus. Okay. In case of prokaryotic cell, ribosomes are present where inside the cytoplasm. So if you see the basic difference between prokaryotic cell and eukaryotic cell with respect to ribosome, okay, this particular prokaryotic cell, it has ribosomes of which type? 70S type. And eukaryotic cell have ribosomes of which type? ATS type. Okay. So if you see over here, this ribosomes, they have two units. One is small unit, one is large unit. Okay. The small unit in case of prokaryotic cell is 30S. And the large unit in case of prokaryotic cell is 50S. Okay, whereas in case of eukaryotic cells, same. The ATS type of ribosome, they have two units, smaller unit and larger unit. Okay, the smaller unit is 40S subunit and the larger unit is 60S subunit. First, understand one thing. In case of prokaryotic cell, which type of ribosome is present? 70S. In case of eukaryotic, which type of ribosome is present? ATS. Now 70S ribosome is combination of what? Small and large subunit. This is small, this is large. Okay. This is the small subunit, this is the large subunit. Okay. So in case of 70S ribosomal arrangement, which two subunits are present? 30S and 50S. Understanding? In case of ATS ribosomal arrangement, in case of eukaryotes, which two subunits are present? What happened to this? Which two subunits are present? 40S and 60S. Okay. So don't add this. 30 plus 50 means 70. No. Over here 40 plus 60 is 100. But still it is 80. Okay. So how we determine this? How this number is determined? Why it is called as 30S? Why it is called as 50S? Why it is called as 40? Why it is called as 60? Okay, understand this thing. This S means what? Sedimentation coefficient. Okay, what do you mean by sedimentation coefficient? Each of the subunit was separated and its sedimentation rate was you can say estimated. Sedimentation rate means what? The rate at which a particular molecule will settle down is called as what? Sedimentation. Now if I give you a mixture of sand and water, okay, you stir it and then you keep it for a while without disturbing. What will happen? The dense particles will sediment at the bottom. The medium density particles will sediment at the medium layer and the very low density particles, they will be suspended in the water. Okay, understanding? So similarly what we do, we take a medium and in that medium what we add this separately. We add this separately. We also separate this and separate this. And we add it in the solution and we see how much time it takes for sedimentation. Now, it's a basic common sense that the proportion of rate of sedimentation is based on what? Based on its weight, molecular weight. Higher the weight, quickly it will sediment. Lower the weight, it will be sedimenting very lessly. It means what? Its density. Understanding? So, after we estimated the sedimentation coefficient, this S means what? It is the parameter which is used to determine the weight, molecular weight and molecular density of that particular subunit. Understanding? So, this S means what? Sedimentation coefficient, which determines the density of that particular subunit. Okay, any problem so far? So over here 70S ribosomes and ATS ribosome. 70S is present in prokaryotes. 80S is present in eukaryotes. Okay. Then we have seen another parameter in the last lecture. The prokaryotic cell does not contain cytoskeleton. Okay. Why? Because their cytoplasm is not large enough to occupy that much amount of cytoskeleton. So another question is, how come then that cell will be having a shape, right? Why is cytoskeleton important? Because that cytoskeleton is going to provide the shape and support to the cell. So that when we say cell is circular, cell is like rectangular, cell is hexagonal in shape, cell is spindle shaped. Okay. So why that particular shape is there? Because of the cytoskeleton. So you must be asking and thinking that if the prokaryotic cell does not have a cytoskeleton, right, then how come it will be attaining a particular shape? Bacteria has a particular shape, right? So even though the prokaryotic cell does not have a cytoskeleton, but it has a cell wall. All bacterial cell, all prokaryotic cell, they have a cell wall. And that cell wall is made up of what? Peptidoglycan. Okay. So that cell wall provides the shape to that particular prokaryotic cell. But in case of eukaryotic cell, who provides the shape to that particular cell? Cell, whereas cytoskeleton is absent in case of prokaryotic cell. Okay, remember this point. Next, the cell division, that is from one cell another cell is formed. This is called as what? Cell division, right? So cell division in case of prokaryotic cell occurs by what? Binary fission. Okay. In case of eukaryotic cell, this cell division occurs by mitosis or meiosis.